Hi everyone, I'm Ethan from 9P and today I'm going to talk about genetic engineering HIV resistant genes from animals into human DNA. Contents. I'll basically introduce what genetic engineering is and how CRISPR-Cas9 works. Then I'll talk about why I chose HIV for this topic, and I'll introduce to you two models. Model 1 is a model that I thought of on my own, and Model 2 is the current model experimented by scientists. I'll talk about their purposes, method, advantages, and disadvantages. Well, so what's genetic engineering? Genetic engineering is basically the process of altering an organism's genome, such as extracting DNA from one organism and combining it into the genome of another individual. So, how does CRISPR-Cas9 work? CRISPR-Cas9 originated from bacteria's defense mechanism against viral DNA. Cas9 is an enzyme called nuclease that cuts DNA. So it basically does the job of a restriction enzyme. It's like a scissor. We can genetically engineer DNA to have the genes we want by using CRISPR-Cas9. And we can either remove single nucleotide bases or entire sections. Therefore, it serves as a potential cure for multiple diseases such as HIV, Alzheimer's, Parkinson's, and even sickle cell disease. Probably cancer too. So how does this work? Basically, as you can see from this diagram, this blue thing is the Cas9 enzyme, the nuclease that cuts DNA. And this guide RNA is very important. This guide RNA tells the Cas9 which sections of DNA they need to remove. The guide RNA corresponds to the DNA that we want to remove. So it's the matching genomic sequence. And this part, the PAM sequence, the yellow part here, the PAM sequence tells where the Cas9 enzyme to lock itself on. It's like a docking bay for the Cas9 enzyme. PAM is short for prototype adjacent motif. The Cas9 enzyme removes DNA and cuts DNA by breaking the sugar phosphate backbone, as a result forming blunt ends in the end. This is the desired DNA we want, and we will insert it to replace the removed DNA. DNA ligase will attach the sugar phosphate backbone together. As a result, a recombinant organism is formed. So I have a brief video.
Nucleus, the resulting complex will lock onto a short sequence known as the PAM.
Ignore the misspell. There are some misspells in the subtitles, but this part is the PAM sequence, PAM. The Cas9 locks itself onto this.
The Cas9 will unzip the DNA and match it to its target RNA.
The target RNA is the guide RNA, and instead of using DNA helicase, Cas9 unwinds and unzips the DNA on its own.
If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA.
So this is basically how Cas9 removes certain DNA sections. So why HIV? HIV is short for human immunodeficiency virus, and it's a worldwide problem, a global epidemic these days. A lot of people have been reported with HIV, and most of them die due to AIDS. Scientists cannot cure HIV yet, but we can delay its symptoms from occurring. Therefore, this shows the importance of how genetic engineering can be used to cure HIV and why they should start finding a solution. It shows the significance of this research. I think curing HIV is rather important, and with CRISPR-Cas9, I believe that scientists have the potential solution for curing HIV. As you can see from this infographic, the number of people living with HIV is constantly increasing, but the people receiving treatment is also increasing too, which is good. But still, these people receiving treatment have a risk of dying. According to this graph from 1990 to 2008, you can see that the number of people living with HIV has increased by an amount of like 26 million people. So you can see how HIV is constantly spreading throughout the years, infecting more and more people. So we should try to find a way to stop HIV from spreading. So purpose. Animals are observed to be only carriers of HIV. However, humans, on the other hand, are sufferers. So I thought, with CRISPR-Cas9, why not extract the sections of genes that gives animals their immunity to HIV and integrate it and combine it with human DNA by using CRISPR-Cas9? So I'll basically explain how. Um, First, you extract the animal's gene that gives them the immunity to HIV with CRISPR-Cas9. In this case, it would be monkeys or apes. We cut out the human gene and we insert the section of DNA that codes for HIV immunity from apes. We use DNA ligase to connect the sugar phosphate backbone, and as a result, the recombinant DNA forms. The next part is rather important because we need to make sure that the entire organism has the immunity to HIV and their offsprings too. So a safe way would be inserting the recombinant DNA into the stem cell or germline cells, such as gametes of sperm and egg, to ensure that the entire individual has the immunity to HIV. Or we could use gene drive. Gene drive mostly guarantees that the particular desired gene will be passed down to almost 100% of offspring. So now I'm going to talk about my DNA model, the model 1. So this is the DNA after we remove a certain section, and this is our desired gene, HIV immunity. Then we connect these two together by connecting their sugar phosphate backbone by using DNA ligase, the red one. This entirely new DNA will be known as a recombinant DNA. Then we insert this recombinant DNA into human embryos or stem cells or germline cells to ensure that the entire organism has immunity to HIV. Then, through cell cycles and DNA replication, this recombinant organism will be immune to HIV. The advantages of this model and CRISPR-Cas9. CRISPR-Cas9 allows scientists to choose whichever section we want to extract or insert by creating our own RNA guide. It's efficient, fast, cheap, and practical on animals. The advantage of this model is that we can cure HIV or other diseases. HIV can stop spreading. The disadvantages is that there's no practical test subject that, like other HIV models I will talk about. There's no practical test subject for scientists to experiment on. Mice are not good subjects for this experiment in this case. However, monkeys are. They are desired subjects because their gene similarity is closest to humans. However, there are some ethical problems. According to animal conservatives, they worry that once gene testing is used on monkeys, then scientists will forever rely on monkeys instead of mice because monkeys are genetically similar to humans. And there are no human test subjects because certain laws prevent it, and a lot of paperwork must be passed before a human can be tested. Last but not least, scientists haven't entirely understood the human genome yet, so they don't know whether the changes in genes can cause unintended consequences. Second model, I'll briefly explain this quickly. Scientists have identified 5 genes essential for HIV infection. The first one is CD4 and CCR5. CD4 is the receptor that allows HIV inside the cell, and CCR5 is basically the same, but it's a receptor found on the surface of T cells. ALCAM is responsible for cell-to-cell HIV transmission to occur. So if this was removed, cell-to-cell HIV transmission will not be able to occur. TPS-T2 and the fifth SEL35B2. We can use CRISPR-Cas9 technology to remove these cells and inactivate T cells. Advantages: we can stop HIV from replicating in one's cells, and it doesn't allow virus to inject genetic material inside the cell. Even if it did, the genetic material wouldn't be able to spread. Disadvantage: it has not been tested on humans like the previous HIV model. Scientists tested it on tissue cultures and it worked, but scientists remain unsure whether these changes in live organisms will have the same effect. And there's no reliable test subject. These are the sources I've used in my research, and thank you for watching. Thank you everyone, and this is the end of my research.